that you need to know for your Step 1 exam. There's a lot of DNA in a cell. In fact, if you stretched all the DNA from a single cell end to end, it would be about a meter long. Since a cell is only a few microns in diameter, it needs a way of packaging all this DNA into a small space. So how does it do this? Well, by coiling the DNA around proteins called histones, which are shown here. There's a few different types of histones, called H2A, H2B, H3, and H4. These aggregate together, and DNA wraps around them twice to form a nucleosome. There's one more type of histone, called H1, which is not in the nucleosome, but along the DNA that links nucleosomes together. So what causes DNA to wrap around histones? This is because the DNA is negatively charged, and histones are rich in the positively charged amino acids, arginine and lysine. The lysines on histones are particularly important because gene transcription is often regulated by covalently adding or removing methyl or acetyl groups on these lysines, which changes their affinity for DNA. Now, the effect of acetylating or methylating amino acids on histones depends on the specific amino acid and the specific histone, and you don't need to know that level of detail for the Step 1. But in general, adding acetyl groups or removing methyl groups allows it to be more accessible to RNA polymerase and other transcriptional machinery. This is known as euchromatin, which you can see here. In the opposite situation, having more methylation or less acetylation results in tighter binding of DNA to histones so that it can't be transcribed, and this is called heterochromatin. So which is transcribed more, heterochromatin or euchromatin? That would be euchromatin, since it's more accessible to the transcriptional machinery. I remember this by thinking that acetyl groups are much larger than methyl groups, and therefore will push DNA away from histones, whereas methyl groups are small and allow it to stay close. That's not actually the mechanism, but it might help you keep these straight. You can also use the mnemonics in the book. Histone methylation mostly makes DNA mute, and histone acetylation makes DNA active. Keep in mind, this is referring to the methylation of the histone proteins, not DNA. Cells can also methylate DNA, specifically at sites called CpG islands, which are just places in the genome enriched for C's and G's. Why is it called CpG? The P represents the phosphate which separates one nucleotide from the next. Specifically, the cytosine and adenine nucleotides would be methylated within CpG islands. Okay, so why would a cell methylate CpG islands? This is just another way that cells can repress transcription. If you know you're not going to need to express a gene for a while, or maybe even ever again for this specific cell, methylating a CpG island and its promoter is a good way to make sure you don't transcribe it by mistake. Alright, here's our first flash quiz. We'll be doing a lot of these throughout the chapter, and if you've seen other chapters already, you've seen these before. But I'm going to ask you a quick question, and I'll give you a few seconds to answer it. Methylation of CpG islands has what effect on transcription? So the answer to this one is it represses transcription. Remember, methylation makes DNA mute, and actually methylation of histones generally represses transcription as well by making them heterochromatic instead of euchromatic. Alright, now let's talk about the nucleotides that make up DNA. There are two types of nucleotides, the purines, which are adenine and guanine, and the pyrimidines, which are cytosine, thymine, and uracil. Uracil is only found in RNA, and thymine is only found in DNA. You can remember these using the mnemonic pure as gold, since the purines are adenine and guanine, and cut the pi, since cytosine, uracil, and thymine are the pyrimidines. Alright, let's see if you're paying attention. Adenine. Is it a purine or a pyrimidine? Right, adenine's a purine. Remember, pure as gold. So fortunately, you don't have to memorize the structures of these for your exam, but there are some features you should be familiar with. First of all, the purines have a double ring structure, whereas the pyrimidines have only a single ring. You can use the cut the pi mnemonic to remember this, because the single ring looks kind of like a pie. The only other things you have to know for the Step 1 are that thymine has a methyl group here, and cytosine can be deaminated to make uracil. So why do we care about all these functional groups anyway? Well, the differences in the locations of the ketones and amine groups determine where and how many hydrogen bonds are made when nucleotides pair up in a double helix. Guanine and cytosine pair up by forming 3 hydrogen bonds, shown by the dashed lines here, whereas adenine and thymine form 2 hydrogen bonds, 1 and 2. So which type of pairing is stronger? That would be the GC bonds, which means it requires more energy to separate DNA strands that are rich in Gs and Cs, compared to those that are rich in As and Ts. This ties into the concept of the melting temperature of DNA, which is the temperature you have to reach to separate 2 strands. DNA is double-stranded at lower temperatures, but as you heat it up, you can get the strands to separate. So would G and C-rich DNA have a higher or lower melting temperature than A and T-rich DNA? G and C-rich DNA would have a higher melting temperature, because again, you need more energy to separate these strands, since they hold on to each other more tightly than the AT bonds. Can you think of other properties of DNA that might affect its melting temperature? One of the other most important ones is DNA length, since shorter stretches of DNA are easier to melt apart. This kind of makes sense, right, since there are fewer total bonds. So who cares how strong DNA bonds are anyway? Well, we'll come back to this in the lab techniques section, since in some types of experiments or diagnostic tests, like PCR, the melting temperature of DNA is very important. And it's also important anytime you have to transcribe DNA into RNA, or replicate DNA, because the proteins that do these things have to first find AT-rich regions so they can separate the strands. Alright, to drive this home, let's do an example. Which of these pieces of double-stranded DNA has a higher melting temperature? I'll give you a couple seconds to think about it. So this would be the bottom strand, since it's both longer and it has more G's and C's, both of which help the strands stay together and increase the melting temperature. Alright, shifting gears a bit, you should also know what components are used to build nucleotides. I'll discuss this more in another slide when we go through the nucleotide synthesis in more detail, but for now, you should know that they're made primarily from amino acids, with purines requiring aspartate, glycine, and glutamine, and pyrimidines requiring aspartate and carbamoyl phosphate. It's convenient that the first letters of the amino acids used to make purines are A, G, and G, since again the purines are adenine and guanine. Alright, the last thing I want to cover here is nomenclature. We've already covered the names of the bases, but nucleotides are more than just bases. They also need to have a sugar and a phosphate. What kind of sugar is it? Well, in RNA it's ribose, and in DNA it's deoxyribose. And what's the difference between these two? In this diagram, you can see that the 2' carbon in the sugar can have either a hydroxyl group, which makes it ribose, or just a hydrogen, which makes it deoxyribose. So when we say a nucleotide, we're talking about the base, the sugar, and one phosphate. If you just have a base and a sugar, that's called a nucleoside. This also affects the names of specific bases. For example, the nucleoside version of adenine is adenosine, and the nucleotide version is adenosine monophosphate. If you add 2 more phosphates to adenosine monophosphate, you get adenosine triphosphate, which as you probably know is the main energy currency of the cell.
Hey everyone, my name's Christina and this video is on de novo pyrimidine and purine synthesis. Imagine you're on your rheumatology elective and about to see a follow-up patient. She's a 49-year-old woman who's been suffering from rheumatoid arthritis for many years. You look back and read up on some of her prior notes and check her medication history. You notice she's been on quite a few drugs. She's currently on methotrexate and a biologic, but she used to be on 6-mercaptopurine and leflunomide in the past. How do methotrexate, 6-mercaptopurine, and leflunomide all treat rheumatoid arthritis? We're going to be going over pyrimidine and purine synthesis. In the next few slides, you should be able to 1. Describe how pyrimidine and purine bases are made, 2. Describe how different pharmaceuticals affect this process, and 3. Recognize the clinical diseases that can be treated with these drugs. Alright, let's get into how purine and pyrimidine bases are made. Why are these pathways so important to know? Well, some of the steps in these pathways are targeted by cancer drugs, since cancer cells need to produce lots of nucleotides in order to replicate. Also, a lot of bacteria have different versions of some of the enzymes involved, which allow us to target them specifically with antibiotics. Recall that purine and pyrimidines are nucleotides. The purines in DNA are adenine and guanine, same as in RNA, and the pyrimidines in DNA are cytosine and thymine, and cytosine and uracil in RNA. De novo synthesis of pyrimidine requires aspartate, while de novo synthesis of purines require aspartate, glycine, glutamine, and THF. Purine-based production can also be synthesized from the purine salvage pathway. First, let's look at the chemical structure of purine and pyrimidine precursors. We'll start with PRPP, or phosphoribosyl pyrophosphate. By the way, what does pyrophosphate mean? It's just two phosphate molecules together. This is going to be one of the many examples in this chapter where you don't need to know the exact molecular structure of something, but being able to appreciate its general properties will help you understand the bigger picture. Okay, so how do we make PRPP? We start with ribose 5-phosphate and the enzyme PRPP synthase, which will take two phosphates from an ATP and add them on, so the ATP becomes AMP and the ribose 5-phosphate becomes PRPP. Now, a nucleotide is a combination of a base, a ribose, and a phosphate, and we have the ribose and phosphate here already. Now, why do we want this pyrophosphate on the other end? It's the same reason that ATP has a few phosphates on it, to create a high energy bond right where we want to add our base. So now when we add the base to the PRPP, which is going to go right on the same carbon that the pyrophosphate is on, we already have the energy to do it. We'll lose these two phosphates when we add the base. Okay, so PRPP is used to make both purines and pyrimidines, but let's start with purines because they're a little bit easier. For purine synthesis, we're going to add a precursor base called hypoxanthine, which looks like the other purine bases, onto our PRPP, which results in a nucleotide called IMP, or inosine monophosphate. From here, modification of functional groups on the base, hypoxanthine, will convert IMP to either AMP and GMP. That's it for purines, not too bad, right? Now let's do pyrimidines. They also use PRPP, and also need another precursor called orotic acid, or orotate. Orotic acid is made from the molecule called carbamoyl phosphate, which is also involved in the urea cycle, which is covered in one of the later biochemistry lectures. The only thing I want to mention here is that if someone is deficient in the enzyme ornithine transcarbamylase, or OTC, they will have a buildup of carbamoyl phosphate, and therefore a buildup of orotic acid as well. This interferes with the body's ability to eliminate ammonia and results in symptoms of hyperammonemia such as tremors or asterexis, slurring of speech, and somnolence, as well as high levels of orotic acid in the blood and urine. Okay, getting back to pyrimidine synthesis. Orotic acid is combined with PRPP with the help of the enzyme UMP synthase in order to form UMP. This enzyme is defective in the disease called orotic aciduria, which causes a buildup of orotic acid that results in failure to thrive, developmental delay, megaloblastic anemia. Unlike ornithine transcarbamylase deficiency, which we just went over, you do not see symptoms of hyperammonemia. UMP can then be further converted to UDP and then CTP. One thing to note is that so far I've only shown you how to make ribonucleotides, which would be used in RNA. What's the only difference between a ribonucleotide and a deoxyribonucleotide? The 2' hydroxyl group, which is only present in ribonucleotides. Deoxyribonucleotides are created using the enzyme ribonucleotide reductase, which removes the hydroxyl group from the second carbon, replacing it with just a hydrogen, which you can see being used to create DUDP from UDP here. Thymidylate synthase is how we make thymidine nucleotides. As you can see from these structures of uracil and thymine, the only difference between them is a methyl group. So we have uracil and we want to make thymine. So how do we add a methyl group? Probably the most common way is using the coenzyme tetrahydrofolate, which in this case donates a methyl group to the uracil to create thymidine. After donating this methyl group, THF becomes dihydrofolate, but the enzyme dihydrofolate reductase restores it back to THF so it can be used again. Since folate is a key part of tetrahydrofolate, people who have a deficiency in folate or folic acid deficiency have defects in the nucleotide synthesis, which in adults can cause anemia, What could happen to a developing fetus with folate deficiency? Well, this can cause neural tube defects such as spina bifida. This is why it's very important for pregnant women or those considering getting pregnant to make sure they're eating green leafy vegetables or taking a folate supplement, which is present in prenatal vitamins. Okay, so this is all very interesting, but why do we need to know about this as a doctor in training? How is this going to show up on our Step 1 exam? Well, it's important that you know how to navigate these pathways and enzymes in order to treat certain diseases. As mentioned earlier, we can treat cancers and bacterial infections by targeting specific steps in the nucleotide synthesis. Mostly cancers, since cancer cells divide quickly and therefore require lots of nucleotides for DNA synthesis, but also bacterial infections, since bacteria mostly use the same pathways as humans, but have slightly different versions of some of these enzymes, allowing us to target them and prevent the bacteria from dividing. What other types of cells divide a lot? Cells in the immune system. So targeting nucleotide synthesis can also be used as a very aggressive immunosuppressant in order to treat inflammatory or autoimmune diseases. Let's go through some examples. Back to this image. There are a few drugs that target dihydrofolate reductase. But what's the difference between them? Well, methotrexate targets a human version, so it's used to treat cancer, and as we briefly mentioned at the beginning of the video, is also a very common drug used to treat RA. Trimethoprim specifically targets a bacterial version of dihydrofolate reductase in order to prevent cellular division in bacteria with having much of an effect on the patient. And pyrimethamine is specific for protozoa. 5-fluorouracil and its prodrug, capecitabine, inhibits thymidine synthase and is used to treat a variety of types of cancers, such as basal cell carcinoma. So here's a question for you. How does taking folate or folinic acid affect the drugs that interfere with this cycle? This is something that always confused me when I was trying to study for Step 1. Folate is often given with 5-fluorouracil because it enhances the binding of the drug to its target enzyme, thymidine synthase, making it more effective. However, in the case of methotrexate and trimethoprim, which normally cause folate deficiency, it's used to reduce side effects, especially in tissues that need a lot of cellular division, such as in bone marrow and gastrointestinal lining. So if this comes up on your exam, in both cases, you do want to give patients folate supplements when you prescribe any of these drugs, but for 5-FU, it's to increase its potency, and for methotrexate and trimethoprim, it's to reduce its side effects. Hydroxyurea inhibits ribonucleotide reductase. And what do you think that's going to do? Ribonucleotide reductase is needed to convert ribonucleotides into deoxyribonucleotides, so again, this prevents DNA synthesis, and this is used in humans to treat cancer. But hydroxyurea has another really important clinical use. Can you recall what that is? It's actually one of the main drugs used to manage sickle cell anemia. How does it help? Well, remember, sickle cell anemia is caused by a mutation in the adult hemoglobin, but hydroxyurea causes these patients to produce more fetal hemoglobin, which reduces the frequency of sickling cells, so this is used prophylactically and as a long-term therapy. You basically start them off on a low dose and increase it as high as they can tolerate without developing cytopenias. Okay, next we got 6-mercaptopurine and azathioprine, which both inhibit purine synthesis. Azathioprine is metabolized into 6-mercaptopurine, and they're both used to treat leukemias, rheumatoid arthritis, inflammatory bowel disease, and SLE. They're also used to wean patients off steroids in chronic disease and used to treat steroid-refractory chronic disease. Leflunomide inhibits an enzyme that converts carbonyl phosphate into orotic acid, which is dihydroorotate dehydrogenase, and essentially inhibits pyrimidine synthesis. It is used as an anti-inflammatory and specifically for rheumatoid arthritis and psoriatic arthritis. The last immunosuppressant here is myclophenolate, and this one is mostly used to prevent rejections in organ transplants. It inhibits IMP dehydrogenase, which converts IMP into GMP. As you can see in the book, there's another drug that targets the same enzyme, ribavirin, but this is used for a very different purpose. As the name may suggest, this is used to treat viral infections such as influenza and hepatitis C, as well as some viral hemorrhagic fevers. Alright, let's do a quick flash quiz. What effect does folic acid supplementation have on 5-FU, methotrexate, and trimethoprim? Recall that folic acid increases the potency in 5-FU and reduces the side effects for methotrexate and trimethoprim. Okay, so the bottom line is, amino acids are the starting point in de novo pyrimidine and purine synthesis. Purine base production, however, can also be synthesized from the purine salvage pathway. Many drugs impact the reactions and enzymes in this process and affects both human and bacterial cells. And lastly, several types of cancers, bacterial infections, and inflammatory and autoimmune conditions are treated by the drugs that act on the pyridine and purine synthesis. Okay, that's all for pyrimidine and purine synthesis. If you like this video, don't forget to give it a thumbs up. Hey everyone, my name's Christina and this video is on purine salvage deficiencies. Imagine you're on your emergency medicine rotation and a well-known patient is being triaged. He's a 4-year-old boy coming in for a suspected acute gout attack. He has been seen in the ED several times for gout and is known to be aggressive. A few of his fingernails are bleeding and he has a healing wound on his forehead. A light bulb goes off in your head. This sounds like Leishmanian syndrome. What is the cause of this disease and how do you treat it? We're going to be going over purine salvage deficiencies. In the next few slides, you should be able to 1. Understand the purine salvage pathway, 2. Describe the different purine salvage deficiencies, 3. Understand clinical significance, and 4. Describe diagnosis and treatment options. First, let's go over how nucleotides are broken down. For the Step 1 exam, it's most important to know how to break down purines. Do you recall what the main building blocks of our purine nucleotides are? To make purines, we start by adding hypoxanthine to PRPP, or phosphorybosyl pyrophosphate, in order to make IMP, which stands for intermediate ribonucleoside monophosphate, though IMP is shorter. HGPRT, which stands for hypoxanthine guanine phosphoribosyl transferase, is an enzyme that catalyzes this reaction. A nice thing about biochemistry is that the enzyme names generally tell you what they do. So in this case, we're transferring a hypoxanthine onto a ribose that has a phosphate on it, which is PRPP. IMP can now be converted into either AMP or GMP. What about the G in HGPRT, which is guanine? HGPRT can do the same thing with guanine as it does with hypoxanthine to make GMP. You need a different enzyme to use with adenine to make AMP, which is APRT, or adenine phosphoribosyl transferase. Okay, so sometimes you don't have enough nucleotides and you want to make more, such in a cell that's rapidly dividing, but sometimes you have too many and you want to get rid of them. So how do we break these guys down? Let's start with GMP. GMP can be broken down into guanosine. Do you remember what the difference is between GMP, guanosine, and guanine? So here's GMP, which as the monophosphate in its name tells you, has one phosphate. It also has a sugar and a base. If we remove the phosphate, we're left with guanosine, and then if we remove the sugar, we're left with the guanine base. We can basically do the same thing to get hypoxanthine back from IMP through inosine. AMP doesn't get converted back to adenosine, instead it gets converted to hypoxanthine through the inosine intermediate. This requires the enzyme adenosine deaminase, or ADA, since we need to deaminate the adenosine to create the hypoxanthine base. So now that we've converted everything into hypoxanthine and guanine, we can now use the enzyme xanthine oxidase, depicted here as XO, in order to convert them first into xanthine and then into uric acid. What happens to the uric acid? It's excreted in the urine. Now that we've got the whole pathway laid out, let's talk about what can go wrong. There are two main purine salvage deficiencies that you need to be aware of for your board exams. One is adenosine deaminase deficiency. What happens if you have a genetic deficiency in this enzyme? Well, if you don't have ADA, adenosine begins to accumulate, and as is often in the case in biochemical pathways, when you have a lot of product, it will inhibit the enzyme that's producing it, causing negative feedback. In this case, it inhibits ribonucleotide reductase. Remember what ribonucleotide reductase does? Easy, just look at the name. Ribonucleotide reductase reduces ribonucleotides, like in RNA, into deoxyribonucleotides, like in DNA, so it's required for the synthesis of all deoxyribonucleotides. Since it's inhibited in the case of ADA deficiency, nucleotides can't be produced, which mostly affects cells that need to divide a lot, such as those in the immune system. Since it's inhibited in the case of ADA deficiency, nucleotides can be produced, which mostly affects cells that need to divide a lot, such as those in the immune system, causing lymphotoxicity. Therefore, patients who have ADA deficiency will have a low lymphocyte count and can also have SCID, or severe combined immunodeficiency disease. SCID is a B and T cell disorder that has two different inheritance patterns. SCID due to ADA deficiency is autosomal recessive in nature, while an X-linked inheritance pattern is due to the defective interleukin-2 receptor gamma chain. It's important to keep these minor differences in check. SCID is also known as a bubble boy disease, which has been popularized in movies and many TV dramas. It results in failure to thrive, chronic diarrhea, and recurrent viral, bacterial, fungal, and protozoal infections. A classic sign is an absent thymic shadow on chest X-ray, which should be in this region here. Diagnosis usually occurs around a few months of age with the clinical presentation of recurrent infections. Screening for SCID is gaining more popularity and is performed routinely in some states through PCR measurements of T-cell receptor concentrations. Screening can also be done if there's a family history of the disease. Treatment involves prophylactic antibiotic treatment, IVIG, and bone marrow transplant for curative measures. You should also make sure to avoid live vaccines in these individuals. The next high-yield disease we'll be going over is Lesch-Nyhan syndrome. This involves a deficiency in HGPRT. What happens if a patient is deficient in HGPRT? They won't be able to make GMP or IMP. Instead, the increased guanine and hypoxanthine will just be broken down into uric acid. And what happens when you have too much uric acid? An accumulation of uric acid causes hyperuricemia and gout, as well as renal stones, and indirectly causes some neurological and behavioral problems, including intellectual disability, aggression, and self-mutilation, such as nail and lip biting, as well as head banging. And another classical board question is, what is the inheritance pattern of this disease? HGPRT is on the X chromosome, so this is an X-linked recessive disease. And you can remember that this gene is involved in purine salvage pathways using the mnemonic, he's got purine recovery trouble. The he part is convenient because as an X-linked recessive disorder, you almost always see it in boys. This is usually diagnosed clinically once a triad of uric acid overproduction, neurological dysfunction, and cognitive and behavioral disturbances occur together. Lesch-Nyhan syndrome should always be differential when a child is engaging in aggressive, self-inflicting behaviors. Diagnostically, you could also do a urine test, which can show an elevated urate to creatinine ratio. Genetic testing for the HGPRT enzyme can be done, but it's not typically routinely performed. Alright, so how do we treat this? One commonly used drug is allopurinol, which inhibits xanthine oxidase. This will decrease the level of uric acid in the blood. Now here's an interesting pearl for you. Xanthine oxidase metabolizes normal purines. It can also metabolize drugs that mimic purines. One example is 6-mercaptopurine, and do you remember what that's used for? 6-mercaptopurine is an antineoplastic that is basically a modified purine, which binds to and inhibits enzymes that normally synthesize purines, thus inhibiting purine synthesis. It's used to treat leukemias and inflammatory diseases like rheumatoid arthritis and inflammatory bowel disease. So let's say you have a patient with Lesch-Nyhan syndrome who you're treating with allopurinol, and this patient is now also diagnosed with a neoplasm that needs to be treated with 6-mercaptopurine. What do you think would happen? Since xanthine oxidase normally metabolizes 6-mercaptopurine, but the allopurinol is inhibiting xanthine oxidase, the patient will not be able to metabolize 6-mercaptopurine as effectively, and therefore it needs a lower dose of this medication. Okay, let's move on to a quick flash quiz. What does adenosine deaminase deficiency cause? This causes SCID, which results in failure to thrive and recurrent infections. Recall that SCID due to ADA deficiency has an autosomal recessive inheritance pattern. Okay, so the bottom line, nucleotide acids are made from freed purine bases in the purine salvage pathway. The two main purine salvage deficiencies are ADA deficiency and Lesch-Nyhan syndrome. ADA deficiency results in SCID, failure to thrive, and an array of recurrent infections. Lesch-Nyhan syndrome causes gout, renal stones, intellectual disability, and many behavioral problems. That's all for purine salvage deficiencies. Thanks for watching. If you enjoyed the video, don't forget to give it a thumbs up.
Next, let's talk about the genetic code. I won't spend a lot of time on this since you probably learned about it in high school, but I'll cover some of the key points that you might see in your exam. So the genetic code is what is used to translate DNA, or more directly RNA, into proteins. This is a codon table, and it shows that each amino acid is encoded by sets of 3 nucleotides called codons, and many amino acids are encoded by multiple sets. For example, proline is encoded by CCU, CCC, CCA, and CCG. This means that the code is degenerate or redundant, since multiple codons can code for the same amino acid. You might notice that of the amino acids that have multiple codons, the codons usually only vary by the last nucleotide, which is why this is called the wobble base. Mutations in these third nucleotides will often not affect the amino acid sequence of the protein, and therefore these are often silent mutations. Another feature is that the code is unambiguous, which means that each codon only codes for one amino acid. Our third feature is that it's commaless or non-overlapping. This is easier to explain with an example. So let's say this is part of an RNA sequence that's being read by a ribosome. After each codon is read, the ribosome moves over by 3 nucleotides, so it reads this one, and then this one, and then this one. No overlapping parts of codons will be read, like this, and no nucleotides will be skipped. And the only exception here is that some viruses can read RNA as overlapping codons. The last feature is that the code is universal. This means that in all species the code is the same. If a codon codes for glutamine in one species, it'll also code for glutamine in another species. An exception to this is in mitochondria, which have their own tRNAs, which in some cases match different amino acids to codons than a nuclear encoded gene. While we're here, I also want to point out the 3 codons that don't code for any amino acid, but signal the ribosome to stop, and these are called the stop codons, which are UAA, UAG, and UGA. These are the only 3 that you might want to memorize.
Hey guys, we're going to be going over DNA replication. Can you think of some problems that cells need to consider when replicating their DNA? Well, cells need to make sure they get exactly 2 copies, which are identical, and they need to make sure replication happens only once per cell cycle and at the right time. And they need to make sure that everything happens quickly enough that rapidly dividing cells won't be held up while they wait for their DNA to replicate. After all, you've got 3 billion base pairs, so you need to be efficient. Cells also need to minimize mistakes, such as single nucleotide changes or expansions of repeated sequences. They need to fix mistakes that occur as well, and deal with the issue of not losing DNA at the end of chromosomes, which I'll get back to in a minute. So where do we start? Well, DNA replication starts at an area called the origin replication, which is the center part here. And how many of these are there? In prokaryotic chromosomes, there's only 1, but eukaryotes have a bunch of them, which makes sense since eukaryotes have a lot more DNA to replicate. At this origin of replication, the 2 strands of DNA separate and form what looks like a bubble, and each end of this bubble is called a replication fork. And as replication progresses, this will expand outward as the new strands of DNA are formed. Okay, so let's look at one of these replication forks. So the blue is the parent DNA, and I'll show the newly synthesized DNA in red. The 2 strands replicate in slightly different ways. One replicates continuously, and the other replicates in smaller pieces. The one on the left is called the leading strand, and the one on the right is a lagging strand. Now, why can't the lagging strand create its daughter strand continuously? Well, this has to do with polarity of DNA. Do you know which end of each strand here is a 5' and 3' end? You might want to pause the video and think about it for a second. So DNA is always synthesized in the 5' to 3' direction, and that's referring to the direction of the strand being synthesized, not the original. So on the leading strand, the new piece of DNA can be synthesized continuously, since the replication fork that it's going towards is at the 3' end of the strand being synthesized, and therefore the origin of replication was at the 5' end. However, the lagging strand has to be synthesized in the opposite direction that the replication fork is moving, so as the replication fork progresses, it has to keep starting over. Do you know what each piece of DNA on the lagging strand is called? That would be an Okazaki fragment. Okay, so let's look at some of the enzymes involved. I'll mostly go in chronological order. The first is helicase. So what does helicase do? It unzips the DNA and allows the bubble to expand. A good way to remember this is helicase halves DNA. Helicase disrupts hydrogen bonds between the two strands. It doesn't need to make or break any covalent bonds. Helicases in general belong to a class of DNA enzymes that function not only in DNA replication, but in DNA transcription, translation, and repair. Bloom syndrome is a rare autosomal recessive disease due to a mutated BLM gene. The BLM gene is a member of the protein helicase family, specifically the REC-Q helicase group, and it's important in maintaining the stability of the DNA during the replication process. Hyperrecombination occurs, which causes an increase in chromosome breakage and arrangement, leading to increased mutations. The disease is characterized by sun sensitivity, stunted growth, facial telangiectasias, and higher incidence of malignancies. Now, Bloom syndrome is a lower yield disease to know for your school exams and board exams, so knowing the details and specifics may not be totally necessary, but knowing a mutated BLM gene is due to a mutated helicase function leading to impaired DNA replication is key. Alright, so since helicase is moving along with the replication fork, something is needed to prevent the strands from re-annealing to one another, which complementary strands of DNA like to do. Do you know what protein prevents this? This is where a protein called single-stranded binding protein comes into play, and it does just that. It's a protein that recognizes and binds to single-stranded DNA to prevent it from binding to its complementary sequence on the other strand. The last proteins I'll mention that help open up DNA are the topoisomerases. These are proteins that regulate the supercoiling of DNA. Regulation of supercoiling is a vital process in cells, and you can inhibit them in bacteria using the antibiotics fluoroquinolones, and this basically prevents bacteria from being able to divide. Okay, so what enzyme is in charge of synthesizing the leading strand? That would be DNA polymerase III, which synthesizes the leading strand continuously toward the replication fork, which again is in the 5' to 3' direction. One thing to keep straight is the differences between DNA polymerases between prokaryotes and eukaryotes, which is something I always have to keep straight. Prokaryotes have 3 DNA polymerases, DNA polymerase I, II, and III. These are the ones that you'll most likely be tested on, which makes sense since we can easily use a prokaryote model, such as E. coli, to study their DNA replication process. Eukaryotes, on the other hand, have several DNA polymerases that all end with Greek letters: beta, lambda, sigma, mu, etc. It is super important to understand this distinction because you can easily be asked a question about DNA polymerase III, for example, and you can quickly rule out any eukaryote options. Okay, so back to DNA replication. So now there's one more thing that needs to be done before we can make the lagging strand, and that's adding RNA primers. There are no DNA polymerases that can initiate DNA synthesis from scratch. They have to build off existing DNA or RNA. So what enzyme can help us here? That would be primase, which can synthesize RNA from scratch. And by that I mean it can add ribonucleotides to their complementary DNA bases and connect them together. Once a primer has been laid down, our friend DNA polymerase III can jump in and start adding deoxyribonucleotides to it just like it did for the leading strand. DNA can only be synthesized in the 5' to 3' direction, so DNA polymerase III starts from the RNA primer and builds towards the 3' end of the new strand. So as DNA polymerase III is making each new strand, it proofreads its work, and if it realizes it made a mistake by adding the wrong nucleotide, it can remove it by using its 3' to 5' exonuclease activity. This is of course the opposite direction as DNA synthesis, since it's going backwards to remove any mistakes. After the lagging strand has been a continuous stretch of alternating RNA primers and Okazaki fragments, the only thing left is for a different polymerase, which is DNA polymerase I, to remove the RNA primer, using its 5' to 3' exonuclease activity, and it then fills in the gap with DNA as it goes. Polymerase 1 has the same synthesis and exonuclease functionality and polarity as polymerase 3, but has the added ability to remove RNA primers. So one last enzyme here, DNase ligase, will come in and seal the holes between the DNA fragments. Keep in mind, this is between fragments of the same strand of DNA, not connecting one strand to its complementary strand in a double helix. The last problem we have is what to do about the ends of the chromosomes. The leading strand has no problem, since it could just synthesize DNA all the way to the end, but the lagging strand is dependent on the RNA primers, and the last RNA primer can't be filled in with DNA once it's been removed, since DNA synthesis would require another primer to build off of. Therefore, a few hundred nucleotides of DNA are lost each time the cell divides. The solution is to use telomerase, which is a ribonucleoprotein that repetitively adds a 6-nucleotide sequence, TTAGGG, or TAG, and it's added to the end of the lagging strand, creating non-coding junk DNA that can be lost without harming the function of the cell. In adults, telomerase is only expressed in cells that need to divide a lot, such as stem cells and other cells in the immune system. Although mutations that cause telomerases to be expressed in other cells are one of the things that are necessary for a neoplastic transformation, since without telomerases, even cancer cells will lose the ability to divide after a while. And that's our video on DNA replication. If you liked the video, don't forget to give a thumbs up.
Hi everyone, this is our lecture on DNA repair, so let's get started. To get a clinical perspective, let's look at a quick flash case. Let's say an 8-year-old girl is brought to the clinic by her parents for a wellness visit after immigrating to the United States 2 weeks ago. While her siblings have clear skin, the girl has freckles and extreme sensitivity to sunlight, developing severe sunburn after only a short period of time. Let's keep this in mind as we go through the lecture. Our learning objectives for this lecture are to compare and contrast various DNA repair mechanisms and to correlate defective DNA repair mechanisms with clinical conditions. Remember, our DNA is in a double helix, so when repairing DNA, we can go about it either as double strand or as a single strand. Our double-stranded mechanisms include non-homologous end joining and homologous recombination, and our single-stranded mechanisms include nucleotide excision repair, base excision repair, and mismatch repair. Let's start with our double-stranded DNA repair first and focus here on non-homologous end joining. Now, non-homologous end joining brings together two ends of DNA fragments to repair double-stranded breaks. So here, let's picture a double-stranded break. Non-homologous end joining will bring together the ends to repair it. Now, I didn't say it was exactly where it was supposed to go. I didn't even say it was the part that broke. It could have been some other double strand laying around. Because in non-homologous end joining, homology is not required. Some DNA may be lost in the process. This repair mechanism is defective in ataxia telangiectasia. The next of our double-stranded DNA repair mechanisms is homologous recombination. As the name implies, it requires two homologous DNA duplexes. As you can see in this image, a strand from damaged double-stranded DNA is repaired using a complementary strand from intact homologous double-stranded DNA as a template. This allows the restoration of duplexes accurately without the loss of nucleotides. This mechanism is defective in breast and ovarian cancers with the BRCA1 mutation and in Fanconi anemia. Now let's move on to our single-strand DNA repair mechanisms and discuss nucleotide excision repair first. Let's look at this image to see what happens. So, some sort of damage, like UV exposure, occurs to damage a nucleotide. What happens next is that specific endonucleases release the oligonucleotides containing the damaged bases, and then DNA polymerase fills the gap, and DNA ligase reseals the gap. This allows us to repair bulky helix-distorting lesions, and this occurs in the G1 phase of the cell cycle. Nucleotide excision repair is defective in xeroderma pigmentosum. In this disease, patients are unable to repair DNA pyrimidine dimers caused by UV exposures, and so they present with dry skin, photosensitivity, and skin cancer. The next of our single-strand DNA repair mechanisms is base excision repair. Let's take a look at this image where it seems one base has deaminated. In this case, a base-specific glycosylase removes the altered base and creates an AP site, which stands for apurinic or apyrimidic, depending on the base. One or more nucleotides are removed by AP endonuclease, which cleaves the 5' end. Then AP lyase cleaves the 3' end. Then DNA polymerase, beta, fills the gap and DNA ligase seals it. I know that sounds like a lot of enzymes that are hard to keep in order, so we do use the mnemonic GEL please to remind us of the order. G for glycosylase to remove the altered base, E for endonuclease, L for lyase, P for polymerase, and L again for ligase. Base excision repair occurs throughout the cell cycle and is important in the repair of spontaneous or toxic deamination. The last DNA repair mechanism we'll discuss is the single-stranded mismatch repair. Like in our last examples, let's take a look at this image to help guide our discussion, where it seems one set is mismatched. In this case, mismatched nucleotides and newly synthesized or unmethylated strand are removed and the gap is filled and resealed. This occurs predominantly in the S phase of the cell cycle and is defective in Lynch syndrome, previously called hereditary non-polyposis colorectal cancer, often abbreviated HNPCC. Lynch syndrome is autosomal dominant and approximately 80% progresses to colorectal cancer. Can you remember which genes are mutated? It's great if you're thinking of MLH1 and MSH2. Great, let's do a quick flash quiz. Which DNA repair mechanism is defective in ataxia telangiectasia? Great if you thought of non-homologous end joining. It's important that you compare the defective DNA repair mechanisms with its affiliated clinical disease. Well, let's return to this case. She seems very photosensitive, even has areas of dry skin on her face. Now, a lot of things can cause photosensitivity, especially certain drugs, but we should also consider the possibility of defective DNA repair causing xeroderma pigmentosum, leaving this patient with the inability to repair DNA damage, like the damage that can be caused by UV light. Which specific repair mechanism is defective if this is the case? Yep, it would be nucleotide excision repair. As always, we need a more thorough clinical history and get more details, but this can certainly be a consideration. Well, we've made it to the end. What's the bottom line? Double-stranded DNA repair mechanisms include non-homologous end joining and homologous recombination. Single-strand DNA repair mechanisms include nucleotide excision repair, base excision repair, and mismatch repair. And defective DNA repair can result in a number of clinical diseases. That's all I have for you here. Please give this video a thumbs up if you liked it, and it'll give you the opportunity to submit a comment if you have any feedback or questions. Thanks for joining me. Study hard.
Hey everyone, we're going to be going over mutations in DNA. DNA replication is pretty efficient, and out of 3 billion base pairs, only a few mistakes are made each time a cell divides. But DNA can be damaged by things like ultraviolet light, X-rays, reactive oxygen species, or chemical mutagens. So how bad is a DNA mutation? Well, that depends on what type of mutation it is. Here's a picture straight from your textbook that I absolutely love because it depicts the different types of mutations so well. But after I started to look at this picture a little bit longer, I realized it may be a little bit confusing if you don't recognize that it's showing the DNA coding sequence and the mRNA codon, but it doesn't show the 3' to 5' DNA template strand. Here's a picture to show what I'm talking about. Some people's brain can automatically go from the DNA coding strand to the mRNA, but my brain never worked that way. I always had to write out the DNA template strand and then form the mRNA codon. So whatever works for you, but I did just want to point this out. Okay, so there are 4 different types of mutations: silent, missense, nonsense, and frameshift mutations. The degree of severity is silent, missense, nonsense, and then frameshift, with frameshift mutations being the most detrimental. So here's a chart that I'm sure all of you are familiar with. You need to memorize everything in this chart: the sequences, the corresponding amino acids, everything. Just kidding, you definitely do not need to memorize anything off this chart, and if anyone tells you otherwise, they're lying. You just have to be able to read it and decipher which amino acid a certain sequence of nucleotides corresponds to. I'll be referring back to this chart quite a bit. First off, let's make sure we know that an amino acid is made up of 3 nucleotides, and the 3 nucleotide sequences are known as a codon. The third position of the codon sequence is known as a tRNA wobble position. A change in the nucleotide in this wobble position will not result in a change in the amino acid the codon codes for. Oh, and by the way, for my people from Louisiana, I went to undergrad there, and anytime I hear the word wobble, a little song starts to play in my mind. So just know, I know. Okay, so let's talk about silent mutations now. Silent mutations are exactly what they sound like, silent, meaning they cause no real change. This is a mutation due to one nucleotide sequence change, and it most commonly occurs in that third wobble position and results in a different codon sequence, but not a different amino acid. Look at the sequence here, which all codes for proline. If we change any nucleotide in that third sequence, it will still result in proline, no matter what. These mutations cause no real damage to our DNA.
Missense mutations are also caused by changing one nucleotide, but in this case, it'll also change the amino acid that the codon codes for. In this example, we're changing the second base from CCU to CUU. This would make an amino acid change from proline to leucine.
This can be relatively harmless or extremely harmful, depending on a few things. First of all, if the mutation is an important part of the protein, such as its active site, even a single amino acid change can prevent some enzymes from functioning. Second, changing from one amino acid to a similar one, such as from a medium-sized nonpolar amino acid to another medium-sized nonpolar amino acid, will usually have a smaller effect than changing a positively charged to a negatively charged amino acid, or vice versa. Lastly, the severity of the mutation depends on the importance of the protein, since some proteins have more vital functions than others. What is the most common disease caused by a missense mutation that you can think of? That's right, sickle cell disease, which also happens to be one of the most commonly tested diseases that you'll see on your Step 1 exam, period. Sickle cell disease is due to a substitution of glutamic acid with valine, which results from a single change in the codon sequence of GAG to GTG. Next is nonsense mutations, which is when the nucleotide change converts a codon that codes for an amino acid to one that codes for a stop codon. Think of the phrase, stop the nonsense. And honestly, I say it in my mind every time and during my Step 1 exam. If we take another look at our codon table, you can see that UUA normally codes for leucine. But if that middle nucleotide is mutated and becomes an A, then we have UAA, which codes for a stop codon. Nonsense mutations have a much stronger effect on protein function since you lose everything downstream of where the mutation occurred, which often has the same effect as deleting the protein entirely. It might be worthwhile to memorize the 3 different stop codons, since there are only 3. I used a phrase that essentially made the 3 stop codon sequence a mnemonic: you go away, you are away, and you are gone, for UGA, UAA, and UAG. Okay, so everything I've talked about so far has been changing one nucleotide base into a different one. Frameshift mutations are unique because it occurs when nucleotides are added or removed rather than just changed. Since this throws off the genetic code, everything downstream from the mutation will code for different and wrong amino acids. Let's look at this example. If we had this sequence broken into these codons by color, we have a sentence, "Can you dig her new hat?" But when we add the letter X right here, all of these codons will be shifted over and will now code for something different. So unlike a missense mutation, where you have one different amino acids, now you have a whole lot of different amino acids. This usually causes a similar amount of damage as nonsense mutations, but in some cases can be dramatically worse. Can you think of why? Well, nonsense mutations usually just result in less effective or non-functional protein, whereas frameshift mutations result in new amino acid sequences which can take on new functions. And these new functions can really throw off cellular homeostasis, potentially leading to cancer or genetic diseases. And in fact, important diseases that are caused by frameshift mutations are Duchenne's muscular dystrophy, Tay-Sachs disease, and familial hypercholesterolemia. The last type of mutation I want to talk about is a splice site mutation, and this is exactly what it sounds like as well, a mutation at a splice site. We'll go through splicing in more detail a little bit later, and if you want, you could go ahead and skip ahead and watch the video before this, but I'll summarize it here. When a gene is transcribed into RNA, the RNA first includes both introns and exons. Which ones are removed? That would be introns, which I remember by thinking that the introns stay in the nucleus, since they're removed while everything else goes out to the cytoplasm to be translated into proteins. But what if we have a mutation right here where this red star is? The splicing machinery needs to recognize very specific sequences to know where to splice. So if one of these sequences has a mutation, splicing won't occur, and the whole RNA will head out of the cytoplasm from the nucleus and be translated into a protein. But now the protein might have another section to it as the ribosome tried to read the intronic sequence. So how severe is this type of mutation? Well, it really depends. For the most part, this will just result in a non-functional protein, either because an extra part prevents it from functioning normally, or because some of that intronic sequence had a stop codon in it, and we'll end up with only one part of the protein being translated. But it's also possible that we could create a new function for this protein, which could have very serious consequences. Most notably, splice site mutations are a rare cause of cancers, dementia, epilepsy, and some types of beta thalassemias. It's especially important to recognize some beta thalassemias are due to splice site mutations because they can describe the perfect beta thalassemia clinical picture of a patient having skeletal deformities, chipmunk facey eyes, and simply show you this picture. And they ask you, what is the disease caused by? Well, you'd have to recognize that these cells are target cells, and the disease you're referring to is beta thalassemia. Then you have to go a step further and recall the cause, which is a splice site mutation. Next, let's go on to a flash quiz. So what are the 4 main types of mutations, and clinically, what diseases are very relevant? So we have silent mutations, missense mutations, nonsense mutations, and frameshift mutations. And again, in summary, sickle cell disease is due to a missense mutation. Duchenne muscular dystrophy, Tay-Sachs disease, and familial hypercholesterolemia is due to a frameshift mutation, while certain cancers, dementia, epilepsy, and some types of beta thalassemia is due to splice site mutations. Thanks for watching. If you liked the video, don't forget to give it a thumbs up.